in different cell types. And that's what we do now know is the truth, but it wasn't known earlier, and it's going to be relevant when I start telling you more about the earlier experiments. So just an example of that would be if you had a single cell, it has exactly the same DNA in both of its daughter cells, but in one daughter cell, a gene that is expressed in, required for neurons would be expressed, versus in another daughter cell, same set of genes, but in this one, an epithelial specific cell is expressed. And that's how these cells take on their fate. And the question about is that expression of that neuron fate, is that a permanent mark? Is that cell capable of actually going back to an earlier developmental fate? And that's something people wanted to address. And so the question of whether the changes that occur in DNA during development as cells differentiate, whether those are irreversible or not. And that's why I have irreversible here in red. So the experiment that was designed to do this is called nuclear reprogramming. And this is something that I'm going to give you a number of experiments that were done with respect to this, but I just want to give you the idea to start. So you have a fertilized egg, and you somehow either remove or destroy its genetic material. So it has no DNA in it, but it's still full of proteins and other molecules. You take that enucleated egg, lacking its nucleus, and you insert into it the nucleus from another cell. And here, this is now the earliest developmental time point. So you take a cell, any cell that is older, because the idea is that as they age, the older they are, the more restricted, and so maybe that DNA is in a form that would not be able to go back to that embryonic state. So if you take a nucleus, and here it says skin, but it could be any cell,